Good day. I'm Funmilola Oluwafeni. I currently work with the Space Agency of Nigeria, National Space Research and Development Agency. The title of the presentation I'll be making is Simulated and Analog Microgravity Platforms, their individual capacities, benefits, and limitations. By way of introduction, the methodology of applying microgravity conditions for specific experimentation depends on the type of research, desired level of gravity, duration of study. And impossible experiments are performed in microgravity environments because of the absence of convection, absence of sedimentation of particles, low shear, capability of supporting three-dimensional tissue formation with ease. The possible samples for research under real microgravity or analog, model, or simulated microgravity are biological organisms such as humans, animals, plants, cells, and microorganisms. And others are micromolecules, fluids, and materials. The general basic principle behind all the ground-based simulators to achieve weightlessness is to have a balance between centripetal force of gravity and centrifugal force of the moving object. This article is an overview of the review of current, real, and simulated microgravity platforms for both short and long-term researchers. Basic simple principle, capability, benefits, capacities, and limitations. The basic principle, capability, benefits, capacities, and limitations of each platform will be stated soon. But there are four classifications. The orbiting microgravity platforms, non-orbiting microgravity platforms, ground-based microgravity analogs, and ground-based microgravity simulators. Under the orbiting microgravity platforms, we have the space laboratories such as the ISS, and then we have autonomous microgravity laboratory platform. Under the non-orbiting microgravity platforms, we have the drop tower, the sounding rocket, and the parabolic flight. Under the ground-based microgravity analogs, we have the long-duration bed rest and immersion. Under the ground-based microgravity simulator, we have the clinostat, the random positioning machine, rotating well vessel, magnetic levitation device, and centrifuge. For the space laboratory, as mentioned, an example is ISS. It has a gravity force G of between 10 raised to the power minus 5 and 10 raised to the power minus 6. It supplies microgravity constantly. Its orbits as over 400 kilometers above the Earth. All research in diverse fields are possible. The limitation is that it has scarce flight opportunities. For the autonomous microgravity laboratory platform, examples are the CubeSat and the GenSat. For the drop tower, the gravity force active for it is 10 raised to the power minus 2 to 10 raised to the power minus 6. It supplies microgravity for 2 to 10 seconds, gets to an altitude of several meters, and gravitropic reactions in fungi and for electrophysiological studies is what it's used for. And the limitation is that it has short study duration. For suborbital rocket, that's the sounding rocket, it has gravity force active for it is 10 raised to the power minus 3 to 10 raised to the power minus 4. Supplies microgravity for up to 15 minutes, can achieve a peak altitude of over 400 kilometers. Possible analysis are membrane transport, signal transduction, pathways, gene expression, cell physiology and morphology, free flow electrophoresis, and other biotechnological experiments. The limitation is that it has short study duration, and the disadvantage is that recovering of the experimental models from remote locations. Parabolic flight in specialized airplanes, the G-force active for this is ranges between 10 raised to power minus 2 to 10 raised to power minus 3. Supplies microgravity for 20 to 25 seconds and maneuvers to reach an altitude of at least 3 kilometers. Possible experiments are on studies of neuronal responses in experimental animals, signal transduction in human immune cells and osteoblasts, and protein crystallization projects. Limitation of it is the hyper-G accelerations interruption of the microgravity phases. For the long duration bed rest, it's an analog to microgravity, provides access for up to several days on human subjects, mostly employed to simulate microgravity effects on various physiological systems, especially on bone studies, muscle, and the cardiovascular system of humans. The challenge is that subjects rarely, if ever, develop motion sickness during or after, constituting a great discrepancy between this analog and spaceflight. For the immersion, this is a model of microgravity. Limitation to water immersion is that there is discomfort and possible risk of long-term skin contact with the liquid environment. A challenge of water immersion is that subjects do not develop motion sickness. Dry immersion could be used for studying the effects of weightlessness, lasting 5 to 7 days, similar to the duration of short-term flights. For the clinostat, this is the grand simulator of microgravity, and the J active for it is ranges between 10 raised to the power minus 2 to 10 raised to the power minus 3. It provides long-term access for several hours or longer, applicable for microbial responses, mammalian cell materials, and fluid behavior in microgravity. And some types of clinostat have been used on plant growth and development. Availability and the simplicity of clinostat are the major reasons why these devices are the most common approach to attempt to simulate altered gravity conditions. Challenge on long-term experiments are that some samples get increased in mass, such as plants. The changing weight distribution causes bending stresses and other non-random mechanical stimulation. For the RPM, which is random positioning machine, this is a grand simulator of microgravity. The force G active for it is 10 raised to the power minus 2 to 10 raised to the power minus 3 and provides long-term access for several hours and longer. Several biological parameters such as microbial responses, plant cells, and mammalian cell cultures can be experimented on it. For rotating well vessel, this is a grand simulator of microgravity also. Gravity force of 10 raised to the power minus 2 to 10 raised to the power minus 3. It provides long-term access for several hours and longer, used increasingly in studies of microbial responses and mammalian cell behavior. It has been used for research on fish. The limitation is that cannot simulate microgravity properly for relative fast cellular and molecular processes. For magnetic levitation device is an alternative to microgravity simulation. Possible samples are diamagnetic materials, water, and biological tissue. These are repelled from magnetic fields. It levitates cellular organelles such as hypocotyls, rhizoids, statoliths, inrods. Seedlings and bacteria are also used as samples on it. It has also been used to research on frog and frog eggs. Limitation is that it can be used for only their magnetic substances. Centrifuge, actually, this provides hypergravity, but it's also utilized to simulate microgravity. as it helps to understand and to detect gravity-related phenomena, thereby contributing to microgravity studies. Large diameter centrifuge (LDC) is able to execute and manage experiments from 1 minute up to 6 months without stopping. Possible samples on LDC facility are plants, cells, small animals, physical, science, and technological experiments. LDC facility is flexible in terms of experiment scenarios, duration, and possible equipment to use. Possible reduced gravity platforms. Successful application of partial gravity simulation can be developed into new avenue of research. There is the potential to use climostats and random positioning machines to simulate partial or reduced gravity conditions found on the Moon and Mars. The simulated Mars gravity of 0.38 G can be used in various biological studies to help prepare for a human mission to Mars. This encourages future exploration of simulated partial gravity for research subjects. The means or the location of some non-microgravity or microgravity simulations research platforms. For the large diameter centrifuge, its type of it is located at the ESA, Netherlands. For the drop tower, it's available at the Bremen, Germany. And 2D climostat is available at NASDAQ, Nigeria. And of course, the International Space Station is also available for microgravity researchers. In conclusion, the number of international collaboration and joint venture programs are increasing to investigate more about cellular processes under microgravity, irrespective of the different test platform being used. Comparative results are always appreciable. It is recommended that climostats and random positioning machines that have the potential to simulate partial or reduced gravity conditions that are found on the Moon and Mars will be very important to develop new knowledge about the biological, physical, and chemical environment at the lunar and Martian G levels of 0.17 G and 0.38 G respectively. It is also recommended that other new analogs, models, and simulators having better benefits and advantages can be developed. More so, the development on random positioning machine engineering makes it suitable for novel applications such as 3D cell culturing and tissue engineering. I want to say thank you for your active attention. I appreciate it.